Our bodies are mostly water, and this water is divided into different compartments or spaces. Knowing how much fluid is in each compartment is important for understanding health and diagnosing certain medical conditions. This table shows how doctors measure the volume of these compartments. Here's a breakdown of each compartment and how it's measured. Total body water. This is the total amount of water in your body. How it's measured, special tracers are used. These are substances that mix evenly with the body's water. Examples include Deuterium Oxide and Tritium Oxide. These are slightly heavier forms of water that can be tracked in the body. Antipyrine, a drug that distributes evenly throughout body water. Extracellular fluid. This is the fluid outside of your cells. It includes the fluid in your blood vessels and the fluid surrounding your cells. How it's measured. Tracers that don't easily enter cells are used. Examples include Sodium-22, a radioactive isotope of sodium. Iothalamate, a substance that's filtered by the kidneys. Thiosulfate, insulin, and sucrose. These are sugars or other substances that stay mainly in the ECF. Intracellular fluid. This is the fluid inside your cells. How it's measured. It's calculated by subtracting the extracellular fluid volume from the total body water. Plasma volume. This is the liquid part of your blood without the blood cells. How it's measured. Radioiodinated albumin. Albumin is a protein found in plasma, and it can be labeled with a radioactive isotope of iodine. Evans blue dye, a dye that binds to albumin and stays in the plasma. PI is a mnemonic and just another way to refer to this method. Blood volume. This is the total volume of your blood, including blood cells and plasma. How it's measured. Chromium-51 labeled red blood cells. Red blood cells are tagged with a radioactive isotope of chromium. Calculation. Blood volume can also be calculated using the plasma volume and hematocrit, the percentage of red blood cells in your blood. Blood volume equals plasma volume / 1 hematocrit. Interstitial fluid. This is the fluid that surrounds your cells, but is outside of the blood vessels. How it's measured. It's calculated by subtracting the plasma volume from the extracellular fluid volume. Why is this important? Changes in these fluid compartments can indicate various medical conditions such as dehydration, a decrease in total body water, edema, an increase in interstitial fluid, swelling, heart failure, can affect plasma volume and blood volume.